This is the most frustrating thing in the bring up chat back. Thank you to anyone with the patience. Which I don't blame anybody who doesn't have the patience. But yeah, we're learning that whatever reason my internet not want to cooperate for whatever reason, and just wants to crap out at random times. And this might be the ultimate test of all. Like, there's literally there's no pattern. That is the most frustrating part. Of like, I can't point to anything at all as to why it's just whatever. All character building exercises. I need as much character as possible. But yeah, Wick, if you're still around, that's what we're learning. We're learning how much of a patient person I am, and that I just have bad internet. And I need to solve these internet problems. My mom is definitely a character. There are reasons why I don't talk to my mom in too much character. That might explain my lack of character. She retained all of it. Oh well, yeah. I know how. Let's just have an idea how it is. Of like, at some point, as much as I value family, I also value my quality of life and well-being over family ties. And that at some point, it's not worth it to maintain connections that aren't contributing positively to your life at all. I already have enough stress with my internet issue. Now to add on to mom issues as well, that's too much for me. Where was I? Right. So yeah, dosing. Probably cut out exactly where I was talking about dosing. I'm like, yeah, typical clinical trials, you know, start with a low dose, scale upwards. On that, yeah. For most clinical trials, you know, conditions are set and they don't change the conditions. And now, as they're alluding to, it might be better to have adaptive trials. Trials where you can adjust things on the fly. So if researchers can enroll more patients with a rare disease in a trial, they can mitigate some of these challenges. An increasingly popular approach to that is through decentralized trials, in which patients participate at various sites scattered around the country or even across the world. Virtual consultations and wearable technology, from a smartwatch to a designed-for-purpose device, would collect data on patients from their homes, reducing the need for in-person visits. I think this is, you know, as much as I'm hesitant about new technologies and ChatGPT, I think technologies like this, of like wearable technology, in the context of a clinical trial, that you can monitor patients pretty much like in real time, or like, you know, doesn't require in-person visits. I think that is where these new technologies hold a lot of promise. You know, gather good data, and maybe more real-time data. Hey, I will totally shake my finger at ChatGPT. No problem. Very, and I think you probably missed it, understandably, but I did cover some research about sort of chat AIs. Which is like, you know, chat AIs being better than actual doctors, at least when it comes to like answering questions, and actually being empathetic in a message. which is a sad state of affairs. So I see utility in this. If AI can replace, you know, unempathetic physicians, great. But yeah, I guess should be obvious that Bill Synthetic is all for synthetics. So, decentralized trials were by necessity popular during the COVID-19 pandemic, but more work lies ahead for this transition. One of the challenges, of course, is that some of the legal framework that we have in place for clinical trials requires patients to come in, maybe even sign a consent form on a piece of paper, not signing it digitally. There are strict notes. So we need to adapt our legislation to be able to do these trials and still be compliant with the ethical requirements and so forth. So continuous data collection. For some rare diseases, treatment efficacy could be determined from real-world data outside of a clinical trial. Real-world data includes everyday metrics such as blood pressure and how much a person moves, as well as data from electronic health records, right? So almost making the case, like if at some point, we get to like fully wearable technologies or technologies integrated into our bodies, would it even be necessary to hold a clinical trial? Oh, like if you can get all your information from like just hacking into or at least consensually hacking into all of the wearable technology that we have in our bodies, of like, you can get all the biomarkers in real time, because like, we have actual some wearable technology or some implant monitoring your biomarkers in real time, similar to like, we now have sophisticated blood glucose monitors, or like automated insulin pumps, or like wearable insulin pumps. So we might get to the point of like, we don't hold the formal clinical trial, like we just start monitoring the patient population, and then like, giving them the therapy, and then monitoring the patient population, and then going from there. I don't know how I feel about that, and that's probably like way ahead, but I think that is potentially pretty cool, or holds promise. Both Marks and Thurstrup note the value and increasing use of real-world data, plus the role of wearable technology in collecting it. Members of the EMA and the Dutch Medicine Evaluation Board recently reported that real-world data and evidence may provide insightful information on medicines, clinical effects to guide regulatory decisions. As rapidly evolving technologies, wearables gathering real-world data have the potential to transform the design and outcomes of clinical research, says Vivek Subbiah, Associate Professor of Investigational Cancer Therapies at the University of Texas, MD, Anderson Cancer Center in Houston. Those data are needed to minimize the gap between drug performance in the controlled environment of a randomized clinical trial versus the real-world setting. Right. And that's where it might hold the most promise, because, generally speaking, in a controlled clinical trial, there's recruitment conditions. And so it's like, some patients are excluded. They're basically curating patients to some extent. And so it's like, maybe, we won't need a clinical trial like for recruitment per se, or like we wouldn't have eligibility conditions for recruitment. We would just like go with the general population, and start monitoring the general population for that specific patient population with their consent. If we get to like fully wearable technology that, can monitor medical information, or relevant medical information. Basically that is kind of like the disconnect, where it certainly happens, where like what holds promise in a clinical trial doesn't always track in the real world, or an actual real world clinical setting, because like you're usually looking at a very specific patient population in a clinical trial, which doesn't actually fully match the wide diversity in an entire patient population for a specific disease or a condition. That's why, let's see if I can maybe put it up. Uh, there are totally right. Where that's why you have articles like this. This is again in relation to my own condition, of like, the evidence that you get in a clinical trial doesn't always translate perfectly to clinical practice. There's a little bit of a disconnect. And so when it comes to like wearable technology, maybe wearable technology might bridge that gap. Like you might be actually getting real world data in a real world setting. Currently, electronic medical records are the main source of real world data, but Subia expects the use of wearables such as smartphones or smartwatches to increase. Real world data collection is going to be more mainstream, and we're just scratching the surface of the unlimited potential of this relevant technology. For the increased use of wearables data, digital characterization and assessment need to be standardized and harmonized across regulators, says Subia. As he points out, most of the current studies using wearable technology were designed in silos by small teams. We need more collaboration, everyone coming together to come up with minimum standards, he says. Real-world data is already playing a crucial part in studies of metachromatic leukodystrophy, a rare inherited disease that causes the buildup of lipids in the nervous system, and that affects 1 in 40,000 newborns. At the San Raffaele Telethon Institute for Gene Therapy in Milan, Italy, Luigi Naldini and his colleagues tested a hematopoietic stem cell-based therapy for children with early symptoms of metachromatic leukodystrophy. Unusually, they also included children who were at risk but had not yet developed any symptoms. Interesting. So maybe like a preventative therapy that has potential ethical implications. Including children without disease poses substantial concerns, but treating only very sick children could mask any efficacy. You're going to test a new treatment on some individuals who are still not so sick, Naldini notes, but we have learned that if you go to the very sick patients, you may never prove efficacy of your drug because the disease has already been so damaging. Right. This is totally, even though, as they say, there's substantial concerns, definitely a known phenomenon where, typically for the more experimental or less proven treatments, you're usually recruiting patients that have, the more severe form of the condition, whatever it may be, because they probably exhausted other treatment options. And so like once they've exhausted, other probably more standard treatment options, they go with the experimental ones. So if they've exhausted all the standard treatment options, they probably have a more severe form of the condition. Hence, they're moving to the more experimental or, therapies with less evidence behind them. And so if you're only recruiting patients with the more severe condition, you might be masking, as they sort of say, masking a therapeutic benefit of like, hey, this therapy might actually be effective, but we're not seeing it because we're only looking at the severe patients or the patients with the severe form of the condition. This is totally, I pretty much always use my own condition as an example wherever I can. Of like, for a certain class of the medications, the biologics for my disease, they are typically reserved patients with a severe form. And they typically have, not the highest of response rate. But you can kind of see that, the remission rates for the biologics for my condition, they're low, or they're typically low. And one of the explanations might be is that they're typically reserved for patients with severe form of the disease. Where again, pretty much like you're going to like the last line of defense. Naldini and his colleagues identified at-risk children from a natural history database built from family histories, genotypes, and other factors. Their hunch that the treatment might not work in very sick children proved right. Luckily, the outcome has been amazing, Naldini says. While the treatment can benefit only the fraction of patients identified early in the disease course, he says, in those patients, the benefit was almost 100%, because there were almost no remaining health issues after the treatment. And so, equitable access. Some rare diseases are more prevalent in low and middle income countries, where treatments are rarely tested. Whatever clinical trials are being conducted, we have to involve people in the population that will require the intervention, says Julie McCanny, an expert in sickle cell disease at Muhimbili University of Health and Allied Sciences, Tanzania. Clinical trials should be run in the countries most affected by the disease being studied, she argues. For sickle cell disease, the main burden of disease is in Africa, but new expensive treatments will probably be unavailable to people with low incomes or in countries with fewer healthcare resources. Right. Where sickle cell disease or sickle cell anemia, it's a genetic condition, the result of genetic mutation, this has a much higher prevalence in individuals who are pretty much descended, or pretty much, African Americans, generally speaking, or individuals descended from Africa, because sickle cell anemia or sickle cell disease, it actually confers protection against malaria or malaria infection, where it's actually advantageous in the context of Africa, where there's much higher malaria rates, to have sickle cell disease, cell anemia, and hence the gene pretty much gets spread in the population. Researchers in Africa face an added challenge in setting up studies for sickle cell disease or any other disease. In the past 10 or 15 years, if we wanted to conduct clinical research, it had to go through institutions in the global north, McCanny says. The implication was that institutions in Africa lack the infrastructure and technology, especially things such as advanced sequencing to run the studies. Although McCanny points out that initiatives such as H3 Africa have significantly improved genomic research capacity, he finds that most funders continue to require a collaborator from a high-income country for clinical trials. McCanny knows that this obstacle can be overcome only through perseverance and by providing a scientific rationale for and actually conducting research in Africa. As she says, you just have to chip away at it. Conducting more clinical trials in Africa, however, is not enough. The results need to benefit the population struggling the most with sickle cell disease, which disproportionately affects people with low incomes, whether in Africa or in high-income countries. We need to make sure that care improves on all levels during the research in a contemporaneous rather than sequential manner, County said. And it's like, yeah, pretty much all of these points raised in this article are good points, especially highlighted in the context of rare diseases, where, just to kind of like wrap up, it's not always possible to do good and rigorous science, because the conditions, as I just highlighted, are not always there, or you might be limited by sample size or access to enough patients to run a good clinical trial. There's potential ethical issues in terms of denying life-saving therapy to a patient population, and so like how do you take that into account, or how do you get a drug or therapy approved if it's impossible to run the gold standard randomized double-blind placebo-controlled trial, because this therapy for this condition, progeria, very rare condition. I think the odds, literally 1 in a million or 1 in 5 million, something to that effect. Like it was approved based off of pretty much a study, this study and a few others, I'm sure. But as they sort of say here, so in this journal, this is the Journal of Neurology. They do a classification of evidence. This study provides class 4 evidence that lonafarnib, pretty much has a benefit. So they're giving it like a rating, the class of evidence, class 4. What does that actually mean? So this is the Journal of Neurology, and they have like their own classification of evidence, although not every journal has that. And it's actually here, I have it up. Classification of evidence scheme, where you have class 1, that randomized controlled clinical trial, the gold standard, pretty much the best class of evidence. And then the lower down you go, pretty much the less rigorous evidence, where you have class 2, it's a randomized controlled clinical trial. Then class 3, all other controlled trials. And then you have class 4, which that's pretty much what this study is rated as. Basically, the lowest class, class 4. Yeah, exactly. It's a low socioeconomic evidence class. It's like studies not meeting class 1, 2, or 3 criteria, including case reports, case series, consensus, or expert opinion. So it's like, yeah, it pretty much like is the worst class. It's like, oh, this evidence doesn't meet class 1, class 2, class 3. The worst class. And yet, this drug was approved. Where research. Lonafarnib is go through the mechanism. But yeah, that has been investigated in a human clinical trial as a treatment for progeria, which is an extremely rare genetic disorder. And it's like, a phase 2 clinical trial was completed in 2012 for progeria patients. So like, this is in the time point of 2012, as in like, is pretty much research like this that got this drug, that got that drug approved. As in, they used the worst class of evidence. Okay. No. Magnolia, that's Magnolia's even. This is a new class. Magnolia's like class 0, 100%. That's indisputable. Magnolia has ORI approval, just as this drug has FDA approval. I'm like, yeah, it got FDA approval based off of like class 4 evidence or similar to class 4 evidence. Where it's like, they used a very small patient population, 26 participants. Like, what is the actual, so it's rare. 1 in 18 million. I believe it's like one of the rarest, genetic conditions, if not the rarest. Where it's like, 1 in 18 million. So it's like, you only have access to so many patients. And they don't live very long lives, sadly, and unfortunately. Where, I think their average age, prognosis, few people with progeria exceed 13 years of age. So it's like a really, sadly, and unfortunately, die young. So it's like super challenging, hold a study for this type of patient population. where it's like, yeah, they only had 26 participants, 2 years. So given the fact, as they highlighted in the Wikipedia article, of like, most don't make it past 13 years of age. A study on the time course of 2 years, that's like, 25% of some patients' lifespan. Again, super challenging. But yeah, frequency of clinical strokes, headaches, and seizures was reduced from pretrial rates. 3 patients with a history of frequent TIAs, which I'm not sure what TIAs stand for. It doesn't really matter, but yeah. Average clinical stroke frequency of 1.75 per year. So like they're basically like looking, like I said, there's no control group or placebo group. All 26 participants, which were progeria patients, they all got the therapy. And then they're just like tracking outcome. They're just like closely monitoring them and seeing how they're doing. Where it's like they're looking at stroke frequency, frequency of clinical strokes, headaches, and seizures, and they're saying that it was reduced. And they're going from before and after. Where it's like, okay, what was their frequency of headaches before they start this medication, and then what's the frequency of strokes also before this medication, and then they start the medication or the therapy, and then are there improvements that we can measure? And again, they're going with a very small patient population. And yeah, unfortunately, and then what do you do where it's like, a significant portion of your patient population might pass away. Again, super challenging, but it didn't stop it from getting approved, which is a good thing because it turns out that it actually appears to be effective. But it's like, yeah, you can't always get up to class 1 evidence. And what do you do at that point? And this holds true with especially when it comes to studying diet interventions, where a lot of people criticize studies about diet, which understandably so, because these are difficult studies to carry out, because you're not locking away people in a lab, you're not controlling all the variables. Even the most rigorous diet studies of like, hey, we're gonna send you food and you eat it. Even then, there's issues with like, well, you might be sending food, but you're not confirming that they're actually eating it, etc., etc. So does that mean that we can never come to meaningful conclusions about diet? I would say no, you can. Totally difficult and very challenging, but you still form meaningful conclusions about diet. Let's say appropriate diet, healthy diet. I think a good classical example is like, smoking, and how do we learn that smoking was, not good for you and associated with a whole bunch of negative health outcomes. It wasn't through, running a randomized placebo-controlled trial of like, intentionally giving people tobacco cigarettes, having them smoke them. It was like through a long course of time, through rigorous epidemiological data, statistical analysis. Like, okay, yes, pretty much, I think everyone now agrees that, at the very least, smoking causes cancer. As in, like, we've established a causal link between smoking tobacco cigarettes and the development of cancer. Again, huge challenge, but not impossible. It just requires, a lot of evidence and good rigorous analysis. Like what was done here, where again, they're pretty much using a small patient sample of patients, and they're zooming in as closely as possible to track things. You're trying to call me on Discord, really? Oh. Good evening. Can you hear me?
Hello?
Yeah, you're live.
Yeah. Am I echoing or no?
Nope. Although the caveat is I might disconnect at any moment, as you might know.
Sure. Okay. So you mentioned the FDA earlier. It's like the cream of the crop, right?
Yeah.
What's the second best?
In terms of class of evidence, or
Yeah, in the world or in the medicine field or whatever.
yeah. So it's probably not going to be. So it's pretty much like a step down of like. So your gold standard is like your randomized, double-blind, placebo-controlled trial. Probably a
No,
step down.
more like the organization, not like the